All right, everyone, good afternoon, and welcome to our first webinar on our breakthrough series. This is Modulating Kinase Signaling in Disease. I'm Isabel Casas. I'm the Publications Director at the American Society for Biochemistry and Molecular Biology, and I'm thrilled to be your moderator today. This webinar is the first of our breakthrough series, and it's sponsored by the Journal of Biological Chemistry and part of our ASBMB journal portfolio. Specifically, this is a JBC webinar and one of our STEAM flagship ASBMB journals, which has been a cornerstone in advancing biological research for over 100 years. We want to remind you that our editorial team is welcoming submissions in this topic, on today's topics, and many more. But before we dive in, I want to give you a quick reminder that you're welcome to submit your questions at any time using the Q&A feature at the bottom of your screen. We'll address them during the dedicated Q&A session after the presentation. Also, this webinar is being recorded, and the recording will be available on the ASBMB website. It is now my pleasure to introduce today's speaker, Dr. Paul Shapiro. Dr. Shapiro is an Associate Professor and Associate Dean of Research and Advanced Graduate Studies at the University of Maryland School of Pharmacy. He's a distinguished scientist and an expert in kinase signaling pathways with a particular focus on their roles in disease development and treatment strategies. His groundbreaking research has contributed significantly to our understanding of cellular signaling and therapeutic interventions. Today, Dr. Shapiro will be sharing insight into his work on modulating kinase signaling and its implications for disease treatment. Without further ado, please join me in welcoming Dr. Shapiro. Paul, the floor is yours.
Thank you, Isabel. And thank you all for attending this first JVC webinar. It's my pleasure to start this series. I consider myself a basic scientist, but I'm going to talk a little bit about how we've taken some basic science ideas and have now moved some discoveries into clinical testing. So there we go. So just as a requirement, I have some speaker disclosure information. So I have an advisory role on a company that is helping develop these products. Royalties from the university and funding is listed here. So my outline today, I want to talk a little bit about the kinase inhibitor landscape. I'm going to talk about how the MAP kinases, the mitogen-activated protein kinases, recognize substrates, and then how we can use that information to translate those interactions into clinical testing. And I'm going to focus today mostly on these two MAP kinase families, the p38 alpha MAP kinases and the extracellular signal-regulated kinases, or ERKs.
Hey, Paul.
Yes.
We cannot see your slides.
Oh, no. Oh, no.
Can you try resharing, please?
I didn't do anything. I am so sorry. Can you see them now?
Yes.
Yes, we can.
Okay, I apologize for that, folks. All right, let's get started again. So you can see the slides, but here's a title. I'm going to talk so I won't repeat what I said before. Having some problems moving these along. There we go. Okay, here's my disclosures. Just get that out of the way. And here is my outline. So today's outline, I want to talk a little bit about the kinase inhibitor landscape, where we're at. I'm going to then talk a little bit about how MAP kinases can interact with substrates and how we can use that information, basic science ideas, to translate into clinical testing. And I'm going to focus on these two that I just indicated, the p38 alpha MAP kinases in terms of inflammatory disorders, and the extracellular signal-regulated kinases, or ERK1 and 2, for proliferative disorders. So just as a review of the kinase inhibitor landscape, this is you probably can't see the words in here, but that's not important. These are the FDA approvals of small molecules that have been targeting protein kinases over the last about 25 years. And there's just two key points that I want to bring home here is that most of these inhibitors target tyrosine kinases, and most of them are targeting oncology or cancers, different types of cancers. And so, and you can see there's about 80 small molecules now from up until 2024. 2024 is not indicated here. But the point of that is that if you look at the phylogenetic tree of protein kinases, they can be grouped as indicated here. Many of us have probably seen these. And most of the kinases consists of serine threonine kinases. And as a result, most of the residues that get phosphorylated in cells are on serines or threonines. And as you can see, however, most of the FDA-approved drugs target tyrosine kinases. And so what I'm going to talk about a little bit next is the type of inhibition that these kinases use to block the kinase activity. And so in this slide here is basically the kinase inhibitor mechanisms of action. So this is a typical structure of a protein kinase where you have an N-terminal lobe, a C-terminal lobe, a region that connects those two lobes called the hinge region, an ATP binding site, three amino acids called the DFG motif that basically change in structure depending on whether the enzyme is active or inactive, and an activation loop. And so most of those kinase inhibitors that I told you, that I showed in the previous slide, are of two classes, the type 1s or the type 2s, in that they bind to the ATP binding site and either block that enzyme in an active conformation, that would be the DFG in, or an inactive conformation, which would be the DFG out. There are some kinase inhibitors that are referred to as type 3. Those bind adjacent to the ATP binding site and prevent catalysis and the transfer of phosphate from ATP onto a substrate. The type that I'm going to talk about in our research are what are referred to as type 4 modulators. I call them modulators because we're not inhibiting the whole enzyme. And these bind to distinct regions that are involved in, say, substrate interactions. And so what are the challenges that we have with kinase inhibitors? So the current issues that with the current inhibitors in the clinic is really lack of durable efficacy. And those are related to a couple of things: on and off target toxicity. Often by targeting the ATP binding site, you're not going to be selective in which kinases you're going to be inhibiting. So you can inhibit kinases that you don't want to inhibit. Sometimes that's not a bad thing. And then the other issue is acquired resistance. Often kinase inhibitors that target the ATP binding site, the cancers in particular will develop a resistance to that and find alternative ways to turn on the signaling pathway. So we were interested in looking at different ways to target protein kinases in a way that would provide more durable efficacy and prevent things like acquired resistance. So just let me go to the next part of my talk, which is the basically how these kinases recognize substrates. And so just as a review, we have the membrane and the receptors that are involved that feed into these signaling pathways, and focusing again on the ERKs and the P38s, these kinases can have hundreds of different substrates. And those substrates can do a number of things. They can regulate proliferation, inflammation, differentiation, etc. Importantly, they also have negative feedback mechanisms that are involved in shutting off the pathway to maintain a balance between turning the signal on and turning the signal off. Sorry, I got a cold this weekend. I apologize. So the most of the current kinase inhibitors target the ATP binding site and shut off basically all of these functions. Our ideas, in contrast, can be modulate these functions in a way that only blocks things that we want to block and preserves things that we don't want to block. So basically, the goal is to target promoting functions that promote proliferation, promote inflammation. You can refer to that as the gas versus the brake, or anti-functions like negative feedback. So how do we do that? The first thing we need to know is how do kinases recognize substrates? So Luke Cantley's group published a paper last year, or in 2023, I should say, that basically described the sequences that are involved in regulating, say, the serine and threonine kinase substrate selectivity. And so they used a peptide mixture that basically randomized the amino acids in the back or in front or in the back of the serine or threonine phosphorylation site. And they analyzed over 300 different kinases to determine substrate selectivity. And they came up with this motif tree. And so they had classifications. They had N-terminal basic residues. That would be this first one here. So the serine threonines, they got basic residues that drive the interactions. They have proline directed. That would be the examples of the MAP kinases. They have negatively charged residues. That would be here, like phosphorylated residues or other acidic residues. But I think what you can see is that each one of these categories have a lot of different kinases. So there's got to be more in determining what is involved in the interactions between kinases and substrates. And so it turns out that many MAP kinase substrates have different docking sites or domains that are involved in kinase recognition. And the first one I want to talk about is the D site. It's also called the D domain or the DEJL, for docking site for ERK or junk LLX motif. And it's basically basic residues followed by a hydrophobic patch here. And you can see from some examples here, transcription factors, and those aren't the only substrates that kinases can regulate, where you have a basic patch, an LXL motif, and some other hydrophobic residues. And this is involved in interactions with the kinase. And in this case, I have ERK and P38, which have what are called D recruitment sites that interact with the D domain substrates. And those are indicated in these structures here. So just for your reference, the red is indicative of the ATP or catalytic site. The yellow is that DFG motif that I indicated before. The cyan is the activation loop, which is involved in upstream phosphorylating kinases that activate these enzymes. And on the purple and the green are these domains that combine to form the D recruitment site. One is called the ED domain, in particular because of these two acidic residues in the P38 MAP kinases that are threonines in the ERK residues. And then this common docking domain or CD domain, which has these acidic residues. And so these acidic residues are interacting with those basic residues on the D sites of substrates. And then the hydrophobic interactions are occurring here in these purple regions. So that's one way in which kinases can determine selectivity between substrates. And there is evidence to say that if you swap out these EDs for these threonines, you can alter substrate selectivity. The other site that I want to talk about on kinase substrates is called the F site. It has a couple names, the F site or the DF site, which is referring to the docking site for ERK or FXF. So this is the motif here that is characteristic of the F site. And here are some substrates that are involved in recognition by the MAP kinases. And you can see the FXF or the XFF motif here. And so this motif on substrates is interacting with a MAP kinase F recruitment site or an FRS. And these green residues are highlighting those residues that make up the F recruitment site. And this study here described how that interaction occurs using structural biology. So the transcription factor ELK, which is a substrate of both ERK and P38, has an FXF motif. The phosphorylation site is about a few amino acids N-terminal to that. That's that serine. And the interactions between the ELK peptide, which is shown as the sticks here, and this F recruitment site, which are residues like these leucines, 235, leucine 198. Those are residues that are highlighted here in the green, methionine, tyrosine, the tyrosine here. The numbering could be off because it's this numbering is according to the human sequence. This numbering was probably according to the rodent, but they're basically identical sequences. So now we know a little bit about how kinases interact with substrates, not only through the residues surrounding the phosphorylation site, but also through these domains that are on the substrates that are involved in recognition by these recruitment sites on the MAP kinases. And so the last part of my talk, I'm just going to talk a little bit about how we've taken some of those ideas on how substrates and kinases can interact and translated them into clinical ideas. And by modulating MAP kinase functions through targeting these D recruitment sites or these F recruitment sites. And so just for your reference, I'll talk a little bit about ERK and a project that we have on preventing smooth muscle cell proliferation related to asthma or lung airway remodeling through developing compounds that target the F recruitment site, a project that I have with a collaborator on myeloproliferative disorders with compounds targeting the D recruitment site. And then I'll end with talking mostly about targeting P38 alpha and the D recruitment site on that enzyme and in treating inflammatory disorders. And this is the project that has been taken into clinical trials. So just as a basic start, how do we design small molecules that interact in these various sites that we're interested in? And as many of you know, computer-aided drug design is a useful tool to help us achieve that goal. And so my long-term colleague, Alex McCrell, who runs our Computer-Aided Drug Design Center here, basically models grooves between docking domains. So in this image here, we have in green and blue are these various residues that make up the D recruitment site. And the purple here is a putative compound that might fit in that region. And so he screens for compounds with the best fit in that region. He can narrow it down to, say, a few hundred compounds. We can select those compounds and do biological testing in the lab. The first project that I'm just going to really briefly talk about is with a colleague, Deepak Deshpande, at Thomas Jefferson University. And one of the goals that we wanted, one of the issues that is involved in, I'm really sorry about this, couldn't shake this cold, is mitigating features of asthma. And one of the main features of asthma is airway remodeling, hyperplasia of smooth muscle cells. And how that occurs is through ERK signaling through transcription factors. And so this is just data showing lung airways, the thickening that occurs in this mouse model, a house dust mite model. So you treat the mice with house dust mite, and after a few weeks, they develop basically features of asthma and airway remodeling, just as in a human. And if you pre-treat the animal with a test compound, you can mitigate that. And so it turns out that this compound that was used in the study targeted a region right next to the F recruitment site. And that's where it was designed to target. And it targeted this region here in red, which is the F recruitment site. And we now have funding to explore that in further detail, to take those compounds, optimize those compounds, and then take them hopefully into clinical testing. The second ERK project that I want to talk about is with a collaborator, Dave Wiest, who's at Fox Chase Cancer Center. And this is a classic example of how ERK can have pro and anti functions depending on its docking sites. And so here, just for references, are ERK protein kinase. And I have, again, the ATP binding site in yellow, the docking sites circled here in green and red. And those correspond to the D recruitment site on the left here, and then the F recruitment site over here on the right. So it turns out that about 80% of all substrates for ERK proteins utilize the D recruitment site. And so what his studies have shown, and what we've shown together, is that the D site substrates are more involved in the progression of the disease, whereas the F site substrates are more involved in attenuating or the break on the disease. So we've published some studies that targeting the D recruitment site with some compounds help mitigate the proliferative aspects of a myeloproliferative disorder that requires a JAK tyrosine kinase mutation that's found in humans. I apologize, folks. And now I'm going to finish up the last part of my talk talking about the studies that we've done targeting the p38 alpha MAP kinase. And this is the one that is now in phase 2 clinical trials. My long-term Jeff Hasday is a clinician who's worked in the intensive care unit, and he's been treating patients with acute respiratory distress syndrome for many years. And his goal was to try to reduce the mortality of patients who come in on respirators and are intubated. and so what we knew that p38 was an important target. p38 inhibitors have been around for 30 years. None of them made it into the clinic. And the problem is because many of them have targeted both the alpha and the beta isoform, which are quite identical, 75% identical, but they have differences. p38 alpha is more involved in the pro-inflammatory signal, whereas the beta is more involved in the cytoprotective aspects. And that's a very general way to describe it, but we'll just leave it at that. But the alpha isoform is the more desirable one. And so there's a lot of efforts trying to identify p38 alpha isoform selective inhibitors. However, alpha also has pro and negative functions. And so what we want to do is target its D recruitment site that's involved in interactions with pro-inflammatory substrates. And it turns out that there was a region near this site, marked in yellow here, that had differences between the alpha and the beta subunit. And what we could do is we could design molecules that targeted the alpha and did not target the beta, and also target the alpha in a way that only prevented certain pro-inflammatory substrate regulation, but preserved other functions such as negative feedback, as I mentioned before. And so basically, again, using a CAD screen, we identified from thousands of compounds and obtained some optimized compounds. And we came up with this molecule, which we called UM101, that was selective for alpha and not for beta. And that was tested by a number of ways. We used a number of biophysical assays to show those differences using things like differential scanning fluorimetry, surface plasmon resonance. And I'll just show you a little bit of data about how we used an NMR spectroscopy technique, saturation transfer difference spectroscopy, that helped show that this compound was selective for alpha, not for beta, and also for the site that it was designed against. And so what this data shows, based on the electromagnetic energies given into the solution, you get these peaks. And these peaks correspond to regions of your compound. And so all the peaks show up for alpha, beta, and gamma at the certain radiation. And then when you do a differential spectra, the peaks that remain indicate that the compound or regions of the compound are close to your target. And so in alpha, you can see the peaks pretty much are retained here, but in beta, they go away, meaning that the compound is not close to the target. In the mutation where mutations in residues in the region where we designed the compound for, they went away too. So suggesting that certain parts of the compound were interacting with p38 alpha and not beta. Now, this doesn't tell us where on, the mutations give some idea, but this is not definitive proof on where this compound is binding on the target. But it is indicating that there is interactions between the target and the compound. So after that, I'm now going to shift a little bit and talk a little bit about our partnerships with industry. So the university here is very good about protecting intellectual property and patent protection. And so when we had these compounds, we went through that process. They're very good at interacting and facilitating interactions with pharmaceutical companies. And we got licensed, the compounds that we developed got licensed by a company out in California called Genie Life Sciences. And Dr. Lal here is the CEO and co-founder. They're a small biotech that is very interested in developing immunomodulators for various clinical situations. And so based on this, and based on our compounds that we developed, what they wanted to do is improve the compounds that we had identified. And we did it by a technique, computational technique called site identification by ligand competitive saturation, or SILCS fragments. This is a technique developed by Alex McCrell in our department. And it basically gives you these colored cages here. And each of these cages represent a different chemical group. And the information you can obtain from these different cages here is different types of targets and compounds that you can develop that will potentially interact with different sites that you may be interested in. And so our compound here, which is you can't see very well, which is over here, there was certain cage and frag maps that were associated with its interaction. And so if you look at this a little bit closer, basically these frag maps, these different colored cages, can indicate potential places in which you can build your molecule and develop a molecule that has a better binding affinity or better interactions with the target. And what you're looking for basically are these negative green numbers, which are basically energy scores that will predict favorable interactions and avoid the red numbers, which are unfavorable interactions. And so these sticks here represent a compound, and these and potential areas in which you can build and add on different types of structures and modify this chemical in order for it to bind better to the target. And that's exactly what we did with our lead compound, which I referred to previously as UM101. And so this is the structure of it here. These are, again, the frag maps for that. And we ended up with this compound, which has a couple names. It was originally called SF7044, but now, since the company has licensed it, it is now called 1124. And this is the structure, and the frag maps are shown there. And this was published just last year describing this. And just a little bit of details on on some of the biological activity of this compound is shown here. So one of the things that we noticed with these modulators is that they improved endothelial barrier function. And that's an important thing to prevent leakage of fluids, say, into the lung. And that's what these data show. And so this is basically a measurement of protein that leaks into the bronchiolar lavage fluid. And so the control is here, and then our parent compound is here, and then our optimized compound, Gen 1124, is shown here. So a reduction in protein that gets that leaks into the lung, meaning that the endothelial barrier is tightening, and that is an interesting finding, because we could apply these to other types of disorders that might involve endothelial barrier function leak and dysfunction. The compound also improves survival in a mouse model of acute respiratory distress syndrome, and that's what the data is showing over here. And so basically, this is a model where mice are made to have a fever, hyperthermia, and then they're given a bolus of lipopolysaccharide, which induces a massive inflammatory response, and most of the animals die after about 3 or 4 days. If you introduce that insult and then about 4.5 and 24 hours later give them a bolus of the drug, you can improve the medium survival significantly, as shown here. Other aspects that I'm not going to go through for the sake of time is that using things like RNA-seq, we've shown that these compounds inhibit pro-inflammatory signals, but preserve anti-inflammatory signals, especially things like negative feedback. And then we've done kinase screens to show few off targets, and these have good pharmacokinetics. We also tested this compound in a H1N1 viral influenza model, where these animals are given an infection at day 0, and then most of these animals will die after 10, or all of them will die after 10 days. But if you give the infection and wait 2 days and give them a treatment and start treating the animals, then you can enhance survival considerably. So this led us to the clinical trials, which are sponsored by Genie Life Sciences, the company that's licensed these compounds. We completed the first clinical trial in 2021. That was a phase 1 clinical trial. We're now in the process of in working on the phase 2. There's 8 locations around the country. The trial is testing with patients that are diagnosed with acute respiratory distress syndrome. You can read through the details here if you wish. And then, I think we're about out of, my time is about up, so I'm just going to summarize real quickly that I think there is potential to identify, and we've shown evidence that we can identify both isoform and function selective modulators of MAP kinases in a way that you can block the signaling events that you want to block and preserve the signaling of events that you don't want to block. All of these, we have various models of in vivo activity and proliferative inflammatory disorders. And we're looking at other disorders. We're looking at traumatic brain injury models. We're looking at inflammatory bowel disorders, and other types of cancers. Not all cancers use the same type of signals through the MAP kinases, and we're exploring how targeting one site might be beneficial for some cancers as opposed to others. There are some limitations here, though. We don't have definitive structural validation these compounds bind to where they're designed to, and that's something that has been challenging, and hopefully we can still work on that. What we are doing, though, is doing structure-activity relationships based on omics data, transcriptome, proteome, and the biomarkers that come from the various compounds. And so if you change some chemical moiety, do you get a different output from the biomarkers? And would that be applicable for some disorder versus another disorder? And then finally, I'm just going to acknowledge all the folks, the collaborators that Steve Fletcher, who I haven't mentioned yet, is our chemist. I've mentioned, I think, everybody else. And then the funding, and then some of the corporate collaborators, Genie Life Sciences, I mentioned. Silks Bio is a company that Alex McCrell started, that has developed some of these techniques for identifying and modifying compounds that target different kinases and other enzymes. So I think with that, I think that is about my time limit. I apologize for my hacking. I'm gonna stop sharing, and I'd be happy to address any questions.
Thank you so much, Dr. Shapiro, for that insightful presentation. We have around 8 to 10 minutes for questions. You still have time if you'd like to submit your questions using the Q&A box. And we have a bunch to start, Paul. Our first question is, what is the best current approach to predicting which kinases might target a protein substrate? Is there a program beyond Cantley's paper?
Well, I think you first start with whether you know if it's a serine threonine or a tyrosine kinase. There are some resources that identify, I don't have handy right now, but I think there are some resources that you can look to evaluate substrates and the kinases that are involved. So I guess your question is more, if you have a kinase, what are the substrates? Is that how I'm hearing the kinase or the question? And you don't know the substrates? I mean, that is a problem because we don't know all the substrates of the MAP kinases. Did I hear that right, Isabel?
So I have a follow-up, yes, but mostly I have a substrate, but don't know the kinase doing it.
Yeah. Well, if you know the phosphorylation site, that would be helpful. And then you can kind of work backwards. Is it proline directed? Is it basophilic? Is it acidophilic? That's how I would probably do it. And then you can test various inhibitors and see, can you block that? You know, there's a number of ATP competitive inhibitors that you could test that would, if you have some set like a cell model, can you block that phosphorylation and then kind of narrow it down from there? Is how I think I would do it.
All right, thank you so much. Our next question, how many domain recognition sites usually does a kinase have? Or do we know?
That is a good question. The two that I mentioned are probably the most well studied. However, I think that there's subtleties within each of those that help determine selectivity. I think there may be some context that we don't quite understand. You know, that's a very good question, and I think it depends on the kinase as well. You know, it is a complicated area. I don't know if I can answer that question clearly, but there's at least two on the MAP kinases that we know pretty much quite a bit about.
All right. Our next question is, how do you get tissue specificity with any of your inhibitors, especially UM101? How does it get to lung, but not other tissues that express P38 isotypes? Endothelial targeting?
Yeah. So I guess, I mean, you know, if you're giving it systemically, you're not selectively targeting it to a tissue. But I think what the goal is that you're targeting activities that are involved in tissue that have clinical implications, you know, inflammatory signals. So, you know, it's the upregulation of these kinases in tissue that might be more susceptible to the modulators. And we're hoping that it's more inert in the kinase in a tissue that the kinase doesn't have, you know, constitutive or high levels of activity. So, I mean, but it is an important question on, you know, how to achieve tissue selective, you know, targeting. But that's a problem with most of these small molecule kinase inhibitors. They're given mostly systemically, and hopefully they have more effects on, say, a cancer cell than on a normal cell. But we know that they don't. We know that they have toxicities in cardiac tissue, and you know, other intestinal epithelial cells that express high levels of certain kinases. So, that's a good question, but it's I don't think we are we're quite there. We in terms of as a research community.
Great, thank you. I think we have room for one more question. Our last question is, in your animal data, how do you account for the off-target effects of the GEN compound as a significant physiological effect and the MAP kinase as being less critical?
So let me see if I understand the question. So it's asking about off-target effects. So in the in vivo studies, so we've done a number of analyses in different species, non-human primates, to look at that kind of that aspect. And from that, it was determined that the safety level, the profile was high enough to take it into humans. So, these are always concerns. But we're not seeing things like liver damage. You're not seeing cardiotoxicity. A lot of the preclinical pharmacology evaluates those toxicities prior to taking it into the human. So, it's still a concern though, off-target effects. And I can say with any small molecule, you don't really know all of the off-target effects that you might have. It's a cost-benefit type of thing where you're trying to get beneficial effects, limit the off-target toxic effects.
Amazing. Well, thank you everyone for submitting such thoughtful questions. And Dr. Shapiro, thank you for your detailed responses. I would like to hand it back to you for any final thoughts or key takeaways before we conclude today's session.
I just want to say thank you to everyone. And again, I apologize. Picked up a cold in Wisconsin where it was -10 below. I don't blame it on the weather though. But I hope I was somewhat clear in my explanation. I'd be happy if anybody wants to reach out to me with further questions on the program. But I just say this is a project done by a lot of people. It's not just me. These types of projects have to include a lot of different expertise that I don't have, and but it's exciting, and that's why we do the things we do, because it's exciting to explore, and you never know where things will go.
That's very true. Well, thank you so much, Dr. Shapiro, for your time and expertise today. And thank you all of you for joining us. Be sure to keep an eye out for upcoming webinars in our Breakthrough series. And we hope to see you again soon in February. So have a great day, everyone. Thank you for joining and have a great rest of your week. Thank you, Dr. Shapiro, again.
Thank you.